Hi, Bells. So this lesson is a follow-up to cell membrane bubble lab, and it's going to help finish off the lesson on the structure of the cell membrane. So if you want, you can take your notes on the page that had the drawing of the cell membrane and a space for notes, or you can take them in your notebook. But this is just going to go over what each station of the bubble lab was supposed to show you. So station number 1 was that meant to show you that the membrane is fluid and flexible. So you probably noticed with your bubble, you were able to move it around and see that you were able to kind of bend the bubble membrane without breaking it. Your cell membrane is the same way. It's very fluid, and you'll see it referred to as fluid a lot. Those phospholipids have the ability to move back and forth without breaking. Secondly, the cell membrane can self-repair. You probably noticed that when you stuck your finger into the cell membrane, you were able to withdraw your finger and the membrane could repair itself. This really allows phospholipids to kind of go back together because they are polar on the heads, and so they're a little bit attracted to each other, and that allows them to cluster when there's a break, and then they repair once the break is official. They come in and they repair themselves. You also probably noticed that once you dipped your finger into the bubble solution, you were able to move through the cell membrane, and that's because like dissolves like. So it kind of was supposed to show you how similar polarity objects, aka non-polar, can move through the cell membrane. But when you didn't do that, it was rejected and it popped. The third station was when you blew a bubble within a bubble, and this was meant to demonstrate the fact that eukaryotic cells do have those membrane-bound organelles. So organelles like the Golgi apparatus, the ER, etc., are actually attached to the cell membrane. So when you blew that mini bubble inside the big bubble, you were able to see that. And there are many of these organelles that include outer membranes, and those are composed of the phospholipid bilayer as well. Station number 4 was meant to show you that the membrane has proteins that perform special functions. So I'm going to show you a video of what would happen at station number 4.
There we go.
You can see here we have the protein, and now we can move any object through the cell membrane.
Thank you. That is so cool. You're sustained.
So here, hopefully you noticed that the membrane does have those proteins to perform special functions. And these membrane proteins are located throughout the cell membrane. These purple guys are what you'll see. We often refer to the cell membrane as having a fluid-mosaic model. So fluid, like we talked about, it can bend and stretch a little bit without getting broken as easily because those phospholipids move back and forth. And then mosaic is meant to be like a mosaic tile art picture where you have a lot of tiles that make up the whole image. Similarly, the fluid mosaic model of the membrane has different things embedded into it. So in addition to the phospholipids, we have proteins and carbohydrates that are embedded into the membrane. The proteins kind of float among the lipids, and then the carbohydrates act as chemical identification cards to help cells identify one another. So in the worksheet I gave you with the picture of the cell membrane, it shows some carbohydrates that have like hello, my name is tags, and also text messages. So those carbohydrates are used to help the proteins send messages to each other by acting as kind of like a flag to say, hey, here I am. I'm this type of cell if you need me or you need to send me a message. So those carbohydrates are really important. But even more important are the membrane proteins. So we have 2 types. The first are membrane-spanning proteins, which are also known as integral proteins. These are permanently connected to the cell and are hard workers, and they, as their name implies, span the entire cell membrane. So you can see here is a membrane-spanning protein, also known as an integral protein. These proteins often function like doors to allow bigger or charged molecules into the cell. Because remember, without them, a lot of times they are not able to pass through. Because they can't get through that hydrophobic layer. So the point of these membrane spanning proteins is they can go all the way through the hydrophobic layer. The second type of protein is called a surface protein, and its other name is a peripheral protein. These proteins can be seen like here or here or here. They can be found on any part of the membrane, either inside or outside of the cell. And they are used for similar things to the membrane-spanning proteins, facilitated diffusion and transport, but they're a little bit weaker and have temporary connections to the membrane. So they can actually move around as opposed to the membrane-spanning proteins, which are permanently attached to the cell. Because they're not as strong and not as permanent, we also have more of them. And the surface proteins are where we often see our carbohydrates attached to. Remember those chemical signals or chemical ID cards, like the glycoprotein or the glycolipid or the carbohydrates, etc. So one question you may want to ask yourself is what the difference is between integral proteins and peripheral proteins. And you also may want to add an integral protein and some peripheral proteins to your drawing of the phospholipid bilayer. Experiment number 5 in the bubble lab was shown to introduce you to something called a gap junction. So I'm going to show you experiment number 5 right now. Here you can see she has a bubble, and she's blowing air into the bubble, and as she pulls the straw, you see that little tiny air. One more time. There you go. So gap junctions are one way that cells can communicate with each other and send materials to each other. In this case, it has to happen between two neighboring cells, but their cytoplasms connect directly and they can move materials from one cell to the other. You can kind of think of this as maybe two cells reaching out their cytoplasm like you would reach out hands with a friend. And then if when you touched hands with your friend, information passed between you or stuff passed between you, that's kind of like what a gap junction does. So it's quick and easy movement from materials from one cell to another as long as the cells are adjacent. Station number 6 of the bubble lab was meant to show a type of prokaryotic cell division called binary fission. And this is when the cell divides in half, including the genetic material. So we'll watch that now.
Go. Nice. Yay. Beautiful.
You can see that the cell is dividing into multiple cells. Animal cells and plant cells, aka eukaryotic cells, have to go through a slightly more complex process called mitosis to divide. But bacterial cells, which also have a cell membrane, can divide in one fell swoop, which is called binary fission. And this occurs when a single-celled bacteria splits into two. That is really the end of the bubble lab and the cell membrane. So if you have any other questions, make sure you jot them down. I would recommend going back and making sure you can answer all of the questions that are asked in this presentation and make sure you fully understand the lab. Thank you for listening.